So as was mentioned, technical difficulties already. So I'm here on behalf of the NIH, and we are rolling out a new funding mechanism that is geared toward biomedical researchers and computational biologists. We want to make sure that you guys have access to cloud computing services and space. So essentially, I would love for all of you to come visit me. I'm going to be at poster P82, and we are looking for applicants starting November 30th. Yeah, so please let me know if you're interested, and I look forward to speaking with all of you.
Thank you, Dr. Quattrochi. It's my distinct pleasure to introduce our next keynote speaker, Professor Chris Burge from the MIT. He's a professor in the Department of Biology and Biomedical Engineering. Chris has been a pioneer in the field of computational biology throughout his career, starting with the times of the hidden Markov model-based gene prediction methods such as GenScan, and then moving on to a variety of different problems, including RNA splicing related problems, and RNA binding proteins more recently, which is what he'll be talking about. Chris has been a valued member of the society and has received several awards along the way, including the Overton Prize from the ISCB. And it's great to have you back here again, Chris. Thanks very much for coming here.
Thanks, Rob, for that really nice introduction, and also to Gustavo and Christina for the invitation to be here at this great meeting. So today I want to tell you two stories from my lab's recent work related to protein-RNA interactions and function of alternative 3' UTRs. But I first want to start by just reminding everyone that in addition to transcriptional regulation that we think of all the time, most human genes are also regulated post-transcriptionally. So we know that in the nucleus, alternative splicing is very, very common and can cause subtle or dramatic changes to protein function. And there's also a lot of regulation post-transcriptionally in the cytoplasm, where various factors bind, often UTRs, and regulate translation, RNA decay, or mRNA localization. And so my lab is interested in the rules and mechanisms underlying post-transcriptional gene regulation, connections between different steps in gene expression, as we'll talk about today, and some of the biological functions of post-transcriptional regulation in development and specific diseases. So the two stories I want to talk to you about today relate to sort of two longstanding mysteries of post-transcriptional gene regulation that I find interesting. And so one is the biological function of this phenomenon of 3' UTR lengthening or shortening, which I'll just review in a moment. And secondly, the puzzle of why many even high affinity binding motifs for RNA binding proteins don't appear to be occupied in vivo. I'm trying to understand that. All right, so first a little bit of terminology for alternative 3' UTR. So there are two common types of alternative cleavage and polyadenylation in mammalian genes. So here you have polyadenylation sites, PASs, and in this very common type called tandem UTRs, you have two poly-A sites with no intervening splicing, and so you produce messages with either a shorter 3' UTR if you use the upstream or proximal PAS, or a longer one if you use the distal PAS. And then in the second type, which is also fairly common, called alternative last exons, you have a poly-A site sort of in the middle of this intron, and so through a combination of splicing and cleavage, you can produce two different messages that have completely different 3' UTRs. And the distal PAS might produce the shorter UTR as shown here, or the longer one. It could be either way. And generally, alternative 3' UTRs are common, probably most human genes, and often the sites of cleavage, alternative sites of cleavage, are conserved between human and mouse. Okay, Several years ago now, a postdoc in my lab, Richard Sandberg, collaborating with a postdoc in the Sharp lab, Joel Nielsen, observed in primary mouse T cells that when you activate these T cells and then profile their alternative isoforms, that there's this large-scale shift toward more expression of the shorter 3' UTR isoforms in this sort of tandem poly-A case. We call this 3' UTR shortening because the UTRs appear to get shorter. And then he looked in a variety of available microarray data sets, found that it varies in different tissues, with the brain, for example, tending to have the sort of the longest or most distal poly-A site usage. And looking across, say, the NCI 60 panel or across different tissues, other cases where we had measures of cell proliferation, we found that those tissues that proliferate a lot tend to express the sort of upstream poly-A site and the shorter 3' UTRs, and those that are terminally differentiated are the other way. They express the long 3' UTR isoforms. Differential 3' UTR expression can affect message stability and translation in luciferase assays, for example. And so following this work, there's been a number of other studies from other labs, very interesting studies about various related phenomenon. The Bartel lab, for example, observed that oncogenes are sometimes activated by switching to an upstream poly-A site that can produce several-fold more protein from the same message. Bintian's lab observed progressive lengthening of 3' UTRs in various cell differentiation systems. Christine Meyer's lab showed that genes that are differentially expressed in different tissues usually have one isoform, and that other genes tend to change their isoform usage between tissues. I could go on, but there are a number of interesting phenomena. But none of these have really addressed the function of 3' UTRs sort of in a large scale directly. And so we decided to address that, and in a collaboration with Dave Bartel's lab, profiled the stability and translational capacity as measured by sort of association with polysomes of different 3' UTR isoforms in a mouse 3T3 system. And what we found, sort of to our surprise, was that overall, the stability of different 3' UTR isoforms from the same gene is highly correlated. There are actually very few genes where those two isoforms differ substantially in stability, and the translation of these different 3' UTR isoforms shown here is actually also very correlated. And so what this says is that alternative 3' UTRs rarely affect either stability or translation. And this was not what we were expecting or hoping. We figured there should be big differences, because when you have a longer or shorter 3' UTR, you typically take out or add several hundred bases of sequence, often highly conserved sequence, often containing microRNA target sites, and so forth, and there should be sort of larger differences in this. But at least in this system, there weren't such large differences. And so that raises the puzzle of, if this poly-A regulation doesn't change stability or translation very often, then what is it for? Why is it so conserved? Why does it shift so strongly in differentiation if it's not doing these two things? Okay, and I'll try to answer that today, or at least provide a partial answer. Okay, so I'm going to tell you a story about the work of a postdoc, Matt Tollifer, and it relates to another possible function of 3' UTRs, which is the control of mRNA localization. And so these are some beautiful images from a paper by Eric Lecuyer, where he imaged the different messages in Drosophila embryos and found, for 5,000 different messages, found almost every message has a different pattern of localization. And the protein localization encoded by that message is usually very close to that of the mRNA. And we know in other systems that localization can be very important. So the localization of the ASH1 message in yeast is required for mating type switching, for example. There's some machinery that localizes that's known. And in a mammalian system, localization of beta-actin mRNA to the leading edge is involved, sort of helps polymerize actin at the leading edge and pushes the cytoplasm out and helps it's important in fibroblast motility, for example. But probably the most prominent role for localization occurs in the nervous system, where you have these long projections, neurites, axons, dendrites, and where it's fairly common for some messages to be transported out to the tips of the neurites, and then the message might only be translated once it reaches the synapse, for example, and that might happen even only in response to stimulation, for example. So localization is known, and in a few cases the function is understood, but Matt wanted to understand it in greater detail. This is Matt here. So he chose this CAD system, which is a mouse CNS-derived cell line, where it's these little round cells that when you withdraw serum, within 24 hours or so, most or all of the cells sprout these neurites. And they express a number of neuronal markers and seem to be a reasonable model for neurite growth and neurite localization. And so Matt's strategy was to sort of explore the possibility that genes with different 3' UTR isoforms, like, for example, this gene might have a blue UTR or a red UTR, and if that blue UTR contained elements that localize it to the neurite, then if we could physically fractionate the neurite, separate them from the soma, we would be able to see that. And so the system he used was this so-called transwell assay, where you plate the cells on these porous membranes that have pores that are big enough for the neurites to grow through, but not for a cell body to pass through. Sort of let the neurites grow there for a while, and then you can carefully scrape off the soma, and then the neurites are left in the membrane. And it's a fairly standard assay in the field. And then so we then perform RNA-seq on both the soma RNA and the neurite RNA. And so doing that, he actually did it not only in CAD cells, but also in a neuroblastoma cell line N2As. And we saw very highly correlated localization ratios, where we define the localization ratio as the expression, say FEKM or TPM in the neurite divided by that in the soma. And so you can see you get a range of about 4-fold up to 4-fold down in terms of a neurite versus soma, and it's highly correlated. So it seems to be sort of an intrinsic property of genes. And we also did compared to primary cortical neurons, where it's also correlated, although not quite as highly. And generally, the expected types of genes are localized to neurites. So mitochondrial, ribosomal protein genes, and as well as a number of sort of synaptic messages. Okay, so then looking at the isoform level, Matt looked at sort of the 4 most common types of alternative isoforms in this system. So alternative first exons, which change the 5' UTR, alternative last exons, we talked about tandem UTRs, and skipped exons, which mostly affect the coding region. And what he observed was that it is fairly common for different isoforms to be differentially localized in the system, but it's especially the isoform pairs that alter the 3' UTR that are the ones that are differentially localized. So the alternative last exons are the most common, as well as the tandem UTRs, suggesting that most of the sequence determinants, since these messages only differ in their 3' UTRs, the determinants of localization must often occur there in 3' UTR sequences. Okay. So that actually wasn't surprising. There's quite a bit of literature that suggests 3' UTRs are important. What was surprising was that when we sort of looked at the direction of bias, that is toward the distal first exon, or the distal last exon, or the inclusion isoform versus the exclusion isoform, et cetera. For most of these events, there wasn't a strong bias for, in fact, for tandem UTRs, for example, not a strong bias for distal versus proximal, or for inclusion versus exclusion in terms of which one has higher inclusion in the neurite versus the soma. But for this alternative last exon class here, Matt noticed that there's a prominent bias for the distal isoform to be the one that's more localized to the neurite. And this was very surprising, since these 2 isoforms have different sequences. Presumably, the determinants of localization could evolve easily in either place, and yet predominantly it was this distal isoform that was localized. So he found that if you fuse the distal or proximal UTR isoform onto a reporter, shown here, where we can look at the protein by GFP sort of as an expression or loading control, and then look at the mRNA by RNA fish. You can see the distal ALE reporter, you get lots of mRNA out here in the neurite, and the proximal ALE reporter, you get a similar level of GFP expression, but very little mRNA out in the neurite. So that these UTRs are often sufficient to confer neurite localization, but that still left the puzzle of why would the genomic location of the UTR, that is distal versus proximal, be related to the sort of subcellular, eventual subcellular localization of the encoded messages. And so we scratched our heads a little bit and eventually came up with the hypothesis that perhaps this is a neuron's way of enabling coordinate regulation of the distal ALEs as a subset. That is, you want to turn them all on at one point, and there's some molecular mechanism for doing that. And so some predictions of this model would be that distal ALEs should shift in expression as a group during development or differentiation. Specifically, when you differentiate neurons, have them go from neural precursors to cells that express, that grow neurites. And that there should be a regulatory program, or perhaps specific regulatory factors that are capable of sort of shifting the many genes either all toward distal or all toward proximal, and that, of course, the activity of these factors should change during development and differentiation. And so we have evidence for the first two of these, at least, which I'll show. So the first thing we did was simply differentiate our CAD cells from that round form to the more differentiated form with neurites, and we did RNA-seq, and we found that indeed in the localized, the genes with the localized distal alternative last exon isoforms are in fact induced by this differentiation. Okay. And we also looked in RNA-seq data from, published data from a variety of other systems where there might be some expectation. And here we looked not only at the ALEs, but also at these tandem UTRs and a couple other types of isoforms. And what we found is that in neural precursor differentiation into neurons, there's a shift toward distal tandem UTR and distal ALE. In cardiac differentiation and CAD differentiation, which I showed, as well as at least distal ALEs in cardiac ventricle development. And then in a system where you're actually reversing, you're sort of de-differentiating cells in fibroblast reprogramming, you actually sort of shift back more toward the proximal isoform in both cases. And you can see that it's usually both tandem UTRs and ALEs that shift in the same direction. And so this says that there appear to be systems in place in various developmental contexts that sort of shift these isoforms one way or the other. Again, not terribly, this has been shown before for tandem UTRs, but also we saw it sort of to a similar extent for the ALEs. And then in cancer, where you often get sort of a de-differentiated state, we see here using some data from TCGA for liver cancer and lung cancer that most cancers shift sort of in this red direction, which is toward increased proximal tandem UTR, so that would be 3' UTR shortening here, or proximal ALE expression, sort of away from the distal isoform. Okay, and then about what factors might be controlling this. So here we turn to some data from our collaborator, Brent Gravely, who, as part of an Encode Phase 3 project, had knocked down, at this point, it was about 100 different RNA binding proteins in K562 cells and HepG2 cells, and profiled mRNA expression by RNA-seq. And so here we could look at the fraction of ALEs that went toward the distal isoform or toward the proximal isoform following each of these knockdowns. And what I've done, these are the different RNA binding proteins, and I've highlighted in red those that are sort of known cleavage and polynylation factors. And what the sort of one that really stuck out to us was this one down here, CFIM25, also known as CPSF5, where when you knock it down, you get this really dramatic shift of the tandem UTRs. They almost all go proximal. It's like 90% or something of the ones that change go proximal, and you get also about 75% of the ALEs go proximal. So this is a factor that can sort of affect a large number of UTRs in a consistent direction. And this factor, in fact, was a recent paper from Eric Wagner's lab in which they observed that also, similarly to these results, that it represses proximal cleavage polymorylation sites, and that when you knock it down or overexpress it, you get both this shift in isoforms, and you also can affect tumor size in glioblastoma in a consistent way, where more proximal isoforms tends to be correlated with increased tumor growth. Okay. All right, so to summarize this half of the talk, I've showed you that distal alternative blast exons are associated with neurite localization. They are coordinately regulated during differentiation and repressed in cancer. I didn't show you that muscle-blind proteins we think are a family of proteins that regulates their neuronal localization. And sort of a take-home from all this is that since ALEs seem to follow the same patterns as tandem UTRs in terms of going distal during differentiation and going proximal during dedifferentiation and tumorigenesis, we think that this nomenclature of 3' UTR shortening, lengthening should probably be revised. It'd probably be more accurate to talk about proximal versus distal poly-A site usage, which would encompass both of those two different classes of isoforms and probably reflects more what's going on in terms of the RNA processing machinery. Okay. All right. So in my remaining time, I wanted to tell you a recent story also led by the same postdoc, Matt Tollifer, with a number of contributions from other lab members, where we asked questions about protein-RNA interactions. And so this is really a fundamental question, given that specific protein-RNA interactions drive the specificity of most aspects of gene expression and particularly post-transcriptional gene regulation. And so there is some structural information for some RNA binding proteins. So here's the RBFOX1 protein bound to one of its favorite RNAs, UGC-AUGU. And what you can see is that this sort of, in this case, the RNA kind of threads its way across this beta sheet, which is the RRM domain of RBFOX. And you can sort of, it sort of seems like, FOX is ideally evolved to bind this thing, and it should bind up, if you express enough FOX, it should presumably bind, all the sites, all the UGC-AUGs in the transcriptome. But when you look in vivo by CLIP, and here we're showing some data from Jean Yao's recent sort of enhanced version of CLIP called eCLIP, and I strongly encourage you to take a look at this. It's published in Nature Methods earlier this year. This is a method that gives you much more diverse libraries and comes much closer to saturating coverage of proteins on RNAs. You see that only about 5%, 5 to 10% of preferred binding motifs, that would be GCAUG for this RBFOX2 protein, are bound in introns, and only about 10 to 15% of those sites in 3' UTRs are bound. And of course, CLIP is not fully saturating, and so we did some calculations to sort of estimate the sensitivity of CLIP using a factor called U2AF2, which binds the 3' splice site, and we assume that it should be there at all 3' splice sites, and so therefore you can sort of calculate, looking at what fraction it's actually detected at, what the sensitivity is in this particular assay, and so these numbers up here, that's fairly, it's a quite rough approximation, of course, but these sort of give you an idea that it can't really be binding more than about 15, 20% of these sites in introns and maybe 40% in 3' UTRs, and so there's lots of perfectly good GCAUGs out there that, and this is in a cell type where RBFOX2 is highly expressed and they're just not bound, and so that's the question that we wanted to address. Why some sites are bound, for example, these two sites might be bound, and these other apparently equally good sites are not bound at all in CLIP. And so there's a number of reasons why this might be in vivo. So it could be that, for example, the RNA binding protein is not expressed in the same part of the nucleus where the where that gene is transcribed, for example, or same part of the cytoplasm, for example. It could be that there's another RNA binding protein that binds nearby and kind of sterically occludes this site, for example. Or it could be that secondary structure forms that involves this motif, and that prevents its recognition by that particular RNA binding protein. And so to study this in more, there might be other possibilities too. We were sort of agnostic about this initially, but we did have some clues from some previous work. So a previous grad student, Jason Merkin, had done a classification of alternative exons in terms of their evolutionary age of alternative splicing. So these ones are alternative spliced in all mammals, and these ones are, say, only in the mouse or constitutively spliced. And what he found was that when you look at RBFOX2 clip data in vivo, that, first of all, exons that don't have a FOX motif, which is the minus here, tend not to be bound at all. Those that do have a motif, though, are not equally bound. The constitutive exons that have a motif are only a small fraction of the bound, maybe 5%, whereas the ancient alternative exons, these ones here, are much more likely to be bound. About 30% of them are bound. And so that suggested that presence of the motif is necessary but not sufficient to get good binding in vivo, and that these ancient alternative exons may have evolved additional contextual features that aid in binding by these proteins. And so the way we approach this question is by what we call a natural sequence version of our RNA bind-and-seq, or RBNS assay. And so here we synthesize a pool of oligos. These are regions of about 110 nucleotides that we designed from flanking alternative exons, either upstream proximal, upstream distal, or downstream. And then we order them by array synthesis. We add a T7 promoter. We in vitro transcribe, and then we have RNA representing these natural sequences. We have a pool of about 12,000, and then we incubate this pool with recombinant tagged RNA binding protein, and then pull down the protein on beads and elute the RNA and make a library and sequence it and look at which of these RNAs are bound by the protein and which are not. Okay. And what we found was that even in vitro, with only recombinant protein and RNA present, that first of all, that binding, you don't get any binding if there's no motif, but that if you have a motif, it's not sufficient to get good binding. Only a relatively small proportion of the total were actually bound, here about 20% for these constitutive exons. But it was a larger fraction for the mammalian-wide alternative exons. So suggesting that whatever features that they have that promote greater binding in vivo also are active at least to some extent in vitro. And in vitro, it's a much more simplified system, and really the only plausible explanation then becomes something to do with RNA secondary structure. Okay. This just shows that our assay is quite reproducible. And so here are sets of exons matched for RBFOX motif count, just again showing that the mammalian-wide exons are more likely to be bound by FOX. We saw a similar thing for another RNA binding protein called muscleblind, or MBNL1. Again, the ancient alternative exons are more likely to be bound by muscleblind, both in vivo and in vitro. But when we looked at a more cytoplasmic protein, this Musashi-1 or MSI-1 protein, we didn't see such a difference. So it suggests that it's not just perhaps sort of a greater accessibility or something like that's evolved. It's perhaps greater accessibility of sites for specific alternative splicing regulatory factors that may have been selected for. Okay. All right, so when you look at these natural sequences that have different numbers of binding sites, you see a shift toward the right. So going from 0 to 1 to 2, you get more binding. But when you look at the mammalian-wide alternative exons, the shift is greater for both FOX and muscleblind. So each site sort of is more likely to give you binding compared to Musashi shown here. So then we asked, what about sort of natural regulation? So here's a data set that we had from a paper by McNutt, where there was differentiation from mouse embryonic stem cells all the way to a cortical glutamatergic neuron and RNA-seq all along the way. And so then we looked at alternative splicing and categorized exons into those that had high monotonicity z-scores as a measure of how monotonically the psi value changes during a time course. So mz equals 0 means it sort of bounces around or doesn't really do anything. Mz large means it consistently goes up, and mz negative means it consistently goes down. And we found that the exons which were bound in vitro, or which had flanking intronic sequences bound in vitro, consistently had larger MZ values. They were much more likely to change in their splicing during this developmental system. Okay. So what about at the level of individual sites? So this would be sort of the ultimate if we could predict which of the thousands of GCAUGs in the transcriptome are actually going to be bound by RBFOX and which are not. So we did this comparison where we looked at the RBNS R value or enrichment value of individual segments, and we compared it to the RBFOX2 clip density. And we saw here a positive correlation. It's not a very enormous positive correlation. It's kind of modest, but it was about 0.45 by Spearman, and it was highly significant because there's quite a large number of exons here. And so this actually compares, it's sort of almost in the same range as the correlation of intronic binding that you observe between clip replicates. So introns are notoriously difficult to clip because they're nuclear, they're short-lived, and this is about as good as you can get by clip, and so we're sort of correlating, almost as well. And so it suggests that the in vitro binding is measuring something that predicts in vivo binding. This is just showing that data about these correlations. So correlations of clip replicates here, correlations of RBNS replicates here, and here's the clip versus RBNS. So it's about as good as we could have hoped to do. But what we'd like to do in the future is apply the same natural sequence RBNS approach to 3' UTRs, because 3' UTR is much easier to clip. The messages are much more stable and abundant, and the correlation of clip replicates is much higher, and so that would give us a bigger dynamic range over which to observe to see whether how well does the in vitro really predict in vivo binding, and what are the exceptions, for example. Okay, so our main hypothesis was that those motifs that are more accessible, that have less base pairing probability, are going to be bound better in vitro and in vivo. And so to help address this, we performed this shape-seq method from Kevin Weeks, where you have a chemical that modifies RNA only if it's accessible, basically. And we found that for several hundred regions that those muscle-blind motifs that were bound had consistently lower base pair probability, that is, were more accessible, less RNA secondary structure. And here's just statistics on that. And so the question, another way to address this would be why some perfectly good muscle-blind motif out there is not actually bound. And so we sort of looked at a bunch of those. Here's an example. So we found here's this myo1b intron. This is a gene that's highly expressed in muscle cells where muscle-blind is expressed as well. And it has a UGCU, which is a good motif for muscle-blind, as well as some perhaps weaker motifs here. And this UGCU is just not bound. There's no clip reads anywhere nearby, and it's very poorly bound in vitro. But when you run it through RNA fold, you see there's quite a bit of secondary structure predicted here, including the stem here that is predicted to occlude the UGCU. So we hypothesized, well, maybe something to do with the secondary structure is blocking binding. And so the approach that Danny, a postdoc in the lab, took was to generate natural 100 nucleotide or so intronic sequences that have this UGCU motif that should bind muscle-blind but doesn't, have that motif intact, but the rest of the sequence randomized. And we introduced about 6% mutations at each of the other positions. We, again, had those synthesized, and then transcribe them, and then we incubate this pool of these randomized RNAs with the protein, pull it down, and so forth. And then we're looking for which mutants are going to be enriched in the bound pool versus the unbound pool. Okay. All right, we call this combinatorial mutagenesis, RBNS. And we analyzed two different sequences here, two different sort of natural intronic sequences that sort of have the right motif but are not, don't appear to be bound in vivo. Okay, oops, I mentioned that already. So we saw that mutation frequencies in a sort of control experiment were pretty much flat across this region, but in a, when we have no protein, but when we add muscle blind, you get much higher mutation frequencies at certain positions and lower at others. And then when we folded, looked at the effects of these mutations on the folding, we saw that in the bound pool, you have substantially lower base pair probability right on top of this UGCU motif right here, and higher probability, base pairing probability upstream here and in certain other regions down here. Okay, so suggesting that the mutations that are favored are tending to alter the structure in a way that sort of makes this motif more accessible. You can also look at pairwise mutations and so forth. And again, the pairwise analysis also tends to suggest that those pairs that produce much less, the ones that produce less base pairing are across the motif are the ones that are favored. Okay, so here, just sort of to make it a little more concrete, this is the RNA fold structure of the Myo-1b, sort of the natural oligo that we were studying, and which is poorly bound in vitro. And you can see that the muscle-blind motif is predicted to be at the base of the stem with 3 of the 4 bases paired. Then if you take the 3 mutations which are most depleted, disfavored in the pull-down, you can see it actually, this motif becomes even more buried in a longer stem here. And then if you take the 3 mutations that are most enriched, that are sort of most favored in the bound pool, and you look at what structure that sequence has, so only 3 bases different, the structure is radically different, and this motif here is now sort of presented in a hairpin loop instead of being buried, so it's totally unpaired. And so this suggests that when alternative exons are evolving and acquiring new binding sites for proteins, that there's actually 2 ways they can do this that may be similarly common. So there will be a pool of sort of latent regulatory capacity. So motifs that could bind that protein but don't because they're protected by an RNA secondary structure. And so that the way you could, if you want to acquire binding by this RBP, there's sort of 2 ways to do it. You can have a mutation that creates a new motif here, or you could have a mutation that changes the local secondary structure to a conformation where this motif is now free to bind. And so to ask how common or how much each of those might influence binding, we sort of looked in our pool at various categories. Like originally, the sort of for some sequences, the original motif was still unpaired and no new motif was present. But for others, the motif had become unpaired. So that's and that gave you increased enrichment. Or in other cases, the new motif was present, and that gave you a similar level of new of enrichment. And then, of course, both gives you more enrichment. And when that new motif itself was unpaired, that gave you even stronger enrichment. And so, sort of looking at these data together, we estimate that creation of a new motif and sort of structural alteration that unpairs an existing motif are roughly equally good ways, and perhaps maybe of comparable frequency in the evolution of new binding affinities. So just to summarize this, I told you that whether binding and regulation occur in vivo can be recapitulated to a substantial extent in vitro using a simplified system with only protein and about 100 nucleotides of RNA. And this suggests that local RNA structure plays a dominant role in determining which of the many possible motifs out there are actually going to be binding by proteins. Of course, there's other contributions as well, but this seems to be one of the more important contributions. And I talked to you about these potential evolutionary implications. Okay, and I just want to mention at the end that we're also involved in this Encode Phase 3 project, which is sort of to create a periodic table of RNA binding proteins using a variety of assays, so our RBNS assay, as well as CHIP and CLIP and knockdown RNA-seq, and these are some of the players in that. I'd be happy to talk to people about that work as well. Okay, and I'll just stop here and thank my stylish lab here. In particular, Matt led a lot of this work, and several other, Danny and Peter and Maria were also involved in a number of the analyses, and our collaborators as well, and funding. So, and thanks for your attention. Manolis?
Hi, Chris. Beautiful work. I have two questions, one on the later part, one on the first part. I'll start with the second one on RNA structure. So my question is, if you look at now if you have a sequence that has multiple possible structures, I'm wondering if the shuttling between the different structures in this sort of thermodynamic ensemble of possible structures, if that actually permits the protein to bind during the periods of transition. And I'm wondering if you have some.
Yes. So it is very common. The thermodynamic states of RNA. It's very common to have multiple states with similar energies, and there is thought to be rapid equilibrium between those. And we use sort of the ensemble methods. The ensemble base pair probability to address that rather than the MFE structure. And so so what we think is going on is that as long as you are in this open conformation enough of the time, half the time or some decent fraction of the time, then the protein will have a chance to bind. And once it binds, assuming if the off rate is slow, it'll stay on there and probably prevent the other structure from forming. And so so the subtleties of how much time you spend in each of these structural classes, combined with the set of RNA binding proteins that are sort of in the vicinity, will then those two together will kind of influence the set of actual structures and that are formed. But it can be subtle, and there may also be other proteins that like RNA helicases that use energy to then change that equilibrium and sculpt it.
So, but my question is more specifically distinguishing between an RNA that folds 80% of the time in a single stable position versus 40% of the time in each of two very stable positions. My hypothesis would be that if you have that 80% split into two, the time spent channeling back and forth, which is perhaps not captured by the RNA ensemble structural predictions, might actually give more opportunities for binding.
Oh, I see. When it's switching from one to another, it'll be even less structured than either one. Yeah, no, that is possible. I mean, that would require sort of a deeper sampling of the conformations and stuff. It's an interesting idea that we haven't really addressed that. But
so
interesting thought.
My second question is about the first part on the alternative last exons, basically. I'm wondering, number one, did you look at that in multiple species? Because in particular in flies, we have this very funny property of this abundant stop codon read-through. And this is strongly enriched for a lot of neuronal proteins. So my question is whether the read-through could actually play some of that role of this alternative splicing of the last exon by sort of introducing a domain that might, play similar roles. So I'm wondering if you looked at.
Yeah, we've only looked in mammals so far at that particular question, but that would be interesting to look in flies. And you can imagine also if you read through, you'll knock off all the proteins in that part. What used to be 3' UTR, once it becomes coding, you'll knock everything off and reset that, which could also be important. So that would be worth looking at if we had the right data, which probably exists. Yeah.
So in flies, it's much more abundant than in mammals. In mammals, you don't see as much read-through. There's maybe 5 or 6 proteins.
Yeah. Okay. Yeah. We'll continue back at MIT. Thank you. Thanks, Manolis. Okay.
You said that you measured the structure using shape-seq.
Yes.
Was it on the in vitro oligos or in vivo?
Yeah, we did it in vitro. We made a pool of about 600 and sort of did a pooled shape-seq analysis in vitro. We haven't done it in vivo yet, but one could do that as well. Yeah.
And did you compare it to predictions from RNA fold? Or
Yes, absolutely. So you can just normally the way you use shape is as a constraint on RNA fold, and so or on a similar software, and so we found actually that the structures were very similar between before and after adding the shape data,
so
and
de novo predictions and
yeah, and I think that's because this is sort of an idealized system where 100 nucleotides is about the size that the thermodynamic programs are best at predicting, and so yeah,
and it is in vitro, so you don't.
and it's in vitro, so it's simpler. Yeah, exactly. So the shape didn't add too much, but it was nice to know that it confirmed the thermodynamic models. Yeah.
Very nice work. I've been staring at post-translational modification data, and some of the most highly post-translationally modified proteins are these RNA binding proteins that regulates their localization and probably binding. Are you taking that into account when you look at differences between in vitro and in vivo?
Sure. Yeah, that's a good point. I mean, our in vitro system involves expression in a bacterial system, and so you don't get many, or if any, post-translational modifications on those proteins, and so and yet the proteins bind kind of the same motif that you see enriched in CLIP, or I mean, we have like dozens of cases where we can compare directly in vitro to in vivo binding, and they're definitely strongly correlated. So, I mean, I think that those modifications may more often influence the localization of the protein or its subsequent, regulatory activity more so than directly the RNA binding. But there could also be cases where it affects RNA binding, and one would have to make your proteins in a different way to get that modification on there. Yeah.
The highly modified proteins in the data I'm looking at were not the same ones you were studying, so it may be sort of a bit of both.
It might vary. Yeah, I'd be curious to hear which proteins you're looking at. Yeah, thanks.
Okay.
Jiping?
Yeah, I'm glad to be waiting the cutoff. So really nice work, Chris. I have a question about the first project, the localization. So do you think the previous studies about in cancer cells, there is a shorter 3' UTR, should also be modified to say this is the proximal one versus the distal one? So would that also affect localization of these transcripts in cancers?
Yeah, so the point about the nomenclature is that it certainly is true that cancer cells have shorter 3' UTRs. There's no doubt about that. It's just that what's causing that, we think, is a decision being made in the nucleus at the level of cleavage to choose the proximal cleavage site, and that decision, if you remember, there was a correlation between effects on tandem UTRs and effects on ALEs, and so we think that those two are just regulated in the same way a lot of times, and so if you say proximal PAS or something, then you should expect both of those two types of isoforms to go in the same way, and it just sort of lets you think about both of those two classes together as part of one phenomenon, maybe.
So do you still think it's leading to shorter UTR, so higher expression, more proliferative?
That was our initial idea, and our initial luciferase assay supported that, and the Bartel Labs luciferase assay supported that. But that was on, it's like half a dozen genes, a very small set, and the larger scale work with Bartel, we saw very little effect. It could be it's because of a different cell type. I'm not sure. And other people have observed. I think since then it hasn't been that consistent. I think the shorter one can be more stable and produce more protein, or not, or the other way as well.
But
also,
does localization have an effect?
It could be that cancer cells, because they're less differentiated, require less localization of their messages, and that has something to do with it. So you're not wasting all that time and energy on that localization that you don't need, or something like that. Maybe, yeah. Thanks, Yuping. Yeah.
Just for getting my knowledge off the mic, I should get 2 questions. No, great work. I was actually striking because in the sort of random mutagenesis assay that you show, it looked like the in vitro binding was actually a limit, upper bound for the in vivo, and so the in vivo could occur essentially at any range between the two. So the top quadrant was completely populated randomly, the bottom quadrant was completely empty, right? So is that because the 100 nuclear, the 100 base that you use essentially may not include other motifs that are outside of them that may then fold back on it and prevent binding?
Sure, yeah, it could be that, right, as you add more flanking sequence, generally as you add more sequence, there's more potential base pairing, and so,
in the end, you say that the in vitro actually is a good model, but it seems like the in vitro seems to be a good model for the upper bound of the binding affinity to these.
yeah, no, that is true, that it tends to, in vitro binding tends to be a little bit higher than in vivo, and it could, yeah, that might be a better way of, yeah, I agree with your characterization there, so thanks for that comment. Yeah.
The ISCB has a token of our thanks for Chris. Since we've done away with the conference bags, now it's a privilege of the keynote speakers to have a conference bag and
this is quite an honor.
Badge here. Thanks very much.
Well, yeah. Thank you.